Hi everyone, I'm Dr. Aniša, your microbiology faculty, and in today's session we'll be discussing about a very dreaded topic in microbiology, that is bacterial genetics. And under bacterial genetics we'll be talking about horizontal gene transfer. And for horizontal gene transfer in this particular session, we have included transformation and conjugation, and the rest of the topic would be covered in subsequent session, right? So for horizontal gene transfer, we all are aware of the different methods by which it can be done. So the very first includes transformation. Then we have transduction. Next one is lysogenic conversion. And lastly, we have conjugation. So to begin with, let us talk about transformation. So what do we really mean by transformation? So from transformation, always take the letter F, which would be included in the mechanism and the definition itself. So transformation, it is random uptake of naked free DNA. So with F, we have used the letter F for naked free DNA from the environment by certain competent bacteria. By competent bacteria, all of these words are very, very important for definition as well as mechanism. And integration of this particular free DNA or the DNA strands into the chromosomes of the chromosome of the recipient bacteria. Into chromosome. Right? So this is the definition, and from the definition itself we'll be deriving the mechanism, and it gets very easy with it. So now we have said that there is a lot of free DNA lying around in the environment. So where does that free DNA come from? So bacteria, it is the bacteria after lysis. Bacteria after lysis releases lot of free DNA into the environment, and it is this free DNA present into the environment which is taken up by certain bacteria, right? So what are these bacteria's competent bacteria we have? This is the recipient cell, so this is the naked DNA which is present in the environment, and there we have the recipient cell. So this cell or the bacteria has very easily taken up this DNA into its cell, right? Uptake has been done. So what happens in the next step? So it is the integration of this particular strand or these particular segments of DNA into the chromosome of the bacteria. And what happens next? So if we look closely, this is the chromosome which was present originally in the bacteria. But in this particular image, can you see that in between we have this purple colored different kind of DNA which is integrated inside the chromosome? So it is this purple colored DNA which has now become integrated with the chromosome. And now this particular cell, this particular bacteria becomes transformed cell. And how has that happened? Because of the integration of this free DNA into its chromosome. So we have two parts in the chromosome. We have the recipient DNA, which was originally present in that particular bacteria. And then we have the integrated donor DNA as well. And then the remaining uncombined DNA is present inside the bacterial cell, which has naturally become degraded. So this is the mechanism of transformation. Now we have been saying that we have certain competent bacteria which are very easily taking up this naked free DNA. So what makes these bacteria competent? Firstly, so these are the kind of bacteria which are presently in their log phase of multiplication. They have log phase of multiplication, right? And they also have certain factors which are known as transformation promoting factors, which are known as the competence factors. These are the competence factors. So these two features are present in those competent bacteria. So these features would be displayed by certain bacteria, and that is why they are readily able to take up that particular naked free DNA from the environment. So what are the examples of these particular competent bacteria? Remember with the mnemonic SHIN. So with S we have Streptococcus pneumoniae. With H and I we have Haemophilus influenzae type B. Then with N stands for Neisseria species. And along with that, we have two more bacteria. We have Acinetobacter as well as Pseudomonas. So these are the bacteria which have the capability of transformation. Very, very important. Right? Now moving on to the next part, which is conjugation. So conjugation stands for a term wherein there is transfer of genetic material from one bacteria to other with the help of formation of a conjugation tube. So there is one channel that is formed between two particular bacteria, and the genetic material gets transferred in that pair. So it is genetic material which gets transferred from one bacteria, say donor or male bacteria, so this becomes F positive, F plus, details of which we'll be discussing in the next slide, to the recipient bacteria with the help of formation of a conjugation tube. This is the very important mechanism for conjugation tube. Now talking about the types of conjugation, we first know that the commonest one is F plus and F minus. We'll understand it in later slides. Then we have H FR, and the last one is sexduction, which is nothing but F prime into F negative, right? So now talking about the very first method, which is the simplest one, that is F plus and F minus mating. So what usually happens? So let's have a look at these bacteria. So this, in the very first image, this is the chromosomal DNA which is present in both of these bacteria. Say this is F1, or we can say F plus, and this is F minus. And how do we define that? So this particular bacteria, number 1 bacteria, has plasmid, has a particular plasmid which codes for a gene which is responsible for formation of 𝗧𝗘𝗙𝗚. And this is the gene which is going to get transferred from one bacteria to other. This particular plasmid is going to get transferred from one bacteria to another. So this is the F plasmid or fertility factor. Fertility factor, which is F plus. So fertility plasmid is present in number 1 bacteria, which is absent in number 2. So that is why we say that this is F positive and the other bacteria it is F negative, because it doesn't have F plasmid in it, right? So what happens now, because we say that this F plus plasmid, it encodes for a gene which is responsible for formation of 𝗧𝗘𝗙𝗚 or the conjugation tube. So what happens next? Now these two, this particular plasmid is going to help these two bacteria come very close to each other. So when bacteria number 1 and 2, they become very close to each other, leads to formation of this particular tube. There is a tube which is formed between the two bacteria. And this particular tube, it is known as the conjugation tube. Conjugation tube. Now, since the conjugation tube has been formed between these two bacteria, then the next step that occurs is the transfer of this genetic material or the fertility factor from bacteria 1 to bacteria 2. See, now what happens in next step? The F plasmid, it starts replicating this one strand of the F plasmid, it starts replicating in rolling mechanism, right? And while replicating, it gets transferred from one bacteria, bacteria number 1, to bacteria number 2. So this genetic material has been transferred. This is replicating. Remember that it is not getting degraded, or strands are not getting less in number, they are not getting less in amount, but they are replicating, right? So the strand gets replicated and it gets very well transferred from bacteria number 1 to bacteria number 2. Now one strand has been transferred here, right? So now this strand also replicates and forms 2 copies. So both of these bacteria now have their separate copies of F plasmid. So now what happens? Since in the very first image we said that it is F positive having F plasmid, and the next bacteria, bacteria number 2, did not have any plasmid. Because of the formation of conjugation tube, the transfer of F plasmid has been occurring from bacteria number 1 and bacteria number 2. And finally, we have both the bacteria having F factor or fertility factor. So now it becomes F positive and F positive, right? So now second bacteria is also having fertility factor, and this now can continue with the conjugation from the rest of the bacteria present, right? So there is simplest method wherein transfer of genetic material which is occurring from one bacteria to another bacteria with the help of a channel or a tube, which is conjugation tube, and the factor which is being transferred, it is the fertility factor. So now F negative has been converted to F positive cell, right? This is the simplest method wherein F negative has become F positive by the transfer of fertility factor. Moving on to the next method, which is HFR conjugation, or it also stands for high frequency of recombination. So most of you would relate to a story that I'm about to tell you. So to remember or to learn what exactly happens in HFR conjugation, remember, if you know, there are some highly fashionable residents in your batches, right? And they feel that they are superior and they tend to give fashion advice to everybody, and they move on giving fashion advice to everybody, but they do not tell everything. They do not tell each and every detail of those advices. They are not going to transfer everything to the rest of the residents, but they'll give some part or some piece of their mind or brain to the rest of the residents. So how are we going to use this story in this particular method? So see, so now certain plasmids, say we have the chromosomal DNA in bacteria 1 and bacteria 2. We have the chromosomal DNA in both of these bacteria, and we have this highly fashionable colorful plasmid, which is the F plus plasmid, right? So now certain bacteria, certain plasmids, they have the ability to integrate themselves into the chromosomal DNA of the bacteria, right? So these plasmids, they get integrated into the chromosomal DNA of the bacteria. Can you see? Suppose this is the chromosomal DNA of the bacteria. This is bacteria 1, this is bacteria 2. And there is some colorful integration that has happened. So now, because of this integration, this particular cell becomes the HFR cell. This becomes the HFR cell. Why HFR cell? Because now they have the more ability to transfer genetic material from one cell to another, which refers to as high frequency of recombination, right? So now these are highly fashionable residents. They have the F plasmid integrated into the chromosome. They feel they're superior, right? And this is the bacteria number 2. Say this is F negative. It doesn't have any plasmid, right? So this is F negative. So now what happens? There is of course formation of conjugation tube, because we are talking about conjugation. So there forms a tube. Now what happens in next step? So remember we said that these residents are going to give fashion advice to everybody around in the batch, but they are not going to transfer or give each and every detail to them, right? So now what happens? In this particular step, there is the, suppose this is, take this gray part as, we consider this gray part as the chromosomal DNA. And the rest of the colored part is of the F plasmid that has been integrated, right? So now from bacteria 1 to to bacteria 2, there is some transfer occurring. So along with some part of the plasmid, some portion of the plasmid, we have certain chromosomal DNA which is also getting transferred to other bacteria, right? But before getting whole of the genetic material transferred from bacteria 1 to bacteria 2, the connection breaks between these two bacteria. So the conjugation tube, it breaks before completing the transfer of whole of the material. So it is not the whole genetic material of the plasmid or the chromosomal DNA which gets transferred, but some is left behind. So now what is happening? In bacteria 1, can we see some part of the plasmid which was integrated, right? And in bacteria 2, as we said, we are considering this gray part as the chromosome from bacteria 1. So in bacteria 2, this is the original chromosome in bacteria 2. So it is the chromosome from bacteria 1 which has been transferred along with some part of the plasmid. So rest part of the plasmid is left behind in bacteria 1. So now, since we said that some part of the plasmid and some chromosome has been transferred, not the whole part, so now instead of becoming F positive, the bacteria number 2, instead of becoming F positive, it remains F negative, since not the whole part of the genetic material has been transferred. So, in spite of getting the genetic material from some bacteria, since not whole of the genetic material has been transferred, it still remains F negative. So this is the HFR cell, this is the FHR cell, but the bacteria number 2, which has some part, it still remains F negative, right? So in this particular conjugation, there is the integration of F plasmid into the chromosomal DNA, and that integrated plasmid is transferred to other bacteria, but not whole, but part of the plasmid gets transferred along with certain chromosomal DNA, right? So this becomes the mechanism for HFR conjugation. Remember, highly fashionable residence with HFR and high frequency of recombination, right? Now coming to the next method of conjugation, it is the F prime conjugation. So this dash stands for F prime conjugation. So now remember that transformation of a F plus cell or F plus plasmid into high frequency of recombination HFR cell, it is a reversible method. It is a reversible method and vice versa. Now for this method, let us see that this is an HFR cell wherein we have the F plasmid which has been integrated into the chromosomal DNA. Now it has decided to disintegrate itself from the chromosomal DNA and it wants to get into its free state. Now after disintegration of this particular plasmid from the chromosomal DNA, what happens next? That this particular plasmid, F plasmid, along with its own properties, it takes along some chromosomal DNA and it gets replicated inside the cell. So now this is the F prime cell. Why do we call it F prime? It can be very well said that it is F positive cell. It is only because of the presence of some chromosomal DNA or the transfer of some chromosomal DNA into the F plasmid while disintegrating, right? So it takes along some part of the chromosomal DNA into the plasmid. So now this becomes F prime cell. right? It has its own chromosomal DNA and it has a plasmid having some chromosomal DNA attached. Now, because of the conjugation is happening in the next step, there is of course formation of sex pilus. Now what happens? There is this bacteria number 2 or cell number 2 which is F negative only, right? So what happens next? For the transfer of genetic material, this F prime cell is not just going to transfer its plasmid part or plasmid DNA, it is transferring some part of the chromosomal DNA as well. So replication is not only happening of the plasmid DNA, it is the chromosomal DNA which was originally present in F prime cell to the other cell. So the other cell not just becomes F positive, this particular cell also becomes F prime. Why it is prime? So this particular conjugation method, it is not considering itself superior, but it is superior. It is originally superior because now it actually has some chromosomal DNA in the plasmid, and this chromosomal DNA has been transferred. That is why these are prime people. These are prime bacteria, right? So remember, so the bacteria which is getting transferred with the genetic material also becomes F prime, right? So it is F prime into F negative conjugation wherein the cell becomes F prime finally. So this is the method of F prime conjugation. So now talking about the role of conjugation, we all know the major indication of conjugation is the transfer of drug resistance, transferable drug resistance, and the bacteriocins, which are nothing but antibiotic-like substances which are produced by some bacteria which have the tendency to kill other forms of bacteria, right? And the bacteriocins which are produced by coliform bacteria are cholecystogenic factors, right? So these are the two important roles of conjugation which is important or has an important role in transfer of drug resistance. So these are two important things, and this was all about the conjugation and transformation along with their examples as well as their mechanism. Thank you.